The overall goal of the following experiment is to use in vivo bioluminescence imaging to study immune responses after implantation of engineered heart tissue, or EHT, in rats. This is achieved by first generating luciferase-positive EHTs. Next, EHTs that stably express luciferase are transplanted into the greater omentum of recipient rats. Then, in vivo bioluminescence imaging can be performed on recipient animals to monitor non-invasive longitudinal EHT survival and rejection. Using this model, the intensity and kinetics of acute rejection, for example, can be correlated with the decline of the observed bioluminescent signal intensity over time.
The main advantage of this technique over existing methods like histology is that daily measurements allow for non-invasive longitudinal assessment of graft survival.
This method can provide insight into the survival and rejection of engineered heart tissues, but it can also be applied to other questions, for example, metabolism of EHTs.
The BLI technique will be demonstrated by Christiane Pahrmann, who's performing this technique on a daily basis in my lab.
Transgenic heart cells are first isolated from rosoluciferase Lu neonatal rats. To generate fibrin-based EHT for implantation purposes, prepare a master mix containing cells isolated from neonatal rosoluciferase Lu transgenic rat hearts, bovine fibrinogen and matrigel, 2-XDMEM and NKM. Prepare agarose casting molds by adding 9 ml of 2% agarose in PBS to each well in 6 well culture dishes, and then placing Teflon spacers into the wells. After the agarose solidifies,